This is so cool. And it's just so nice. I'm sure people joke about this, but it's so nice to not have to explain what a synchrotron is in a talk. So looking forward to just telling you, actually, this is gonna, I wrote kind of a different talk for this because I really wanted to talk about some of the techniques that I've been using since graduate school. So I'll be talking about some of the things that me and my PhD advisor, Ninian Blackburn, developed together, and then go into some of the work that I've been doing at Reed that extends upon that. My current work, which is funded by an NSF career, is actually looking at tellurium proteins. And it's not quite ready for prime time in terms of X-ray science yet. So I'm going to really tell you about sort of what I would, what I would be applying to that stuff later. But this is really what I'm about, is using EXAFS to track metal transferrin proteins. So for those of you who mostly deal with non-living things, biochemistry and spectroscopy is just a beautiful combination. So here I have a crystal structure of a protein I began my graduate career with, copper A. It's a dicopper site that has mixed valence. So you can see there's these two coppers in this protein. And basically they actually share some covalency. They act as a little superconductor for electrons in the electron transport chain, which all of us need in respiration. And so you can do all sorts of things with this. You can do crystallography. You can do UV visible spectroscopy, so you can see lots of cool electronic features. And then we have EPR, electron paramagnetic resonance, that you can do with copper. And then you have, of course, extended X-ray absorption fine structure. So I'm going to talk more about that. But the point is, that's what really attracted me to biology, I think, because I was really, as a chemist, interested in lots of different things. But this idea of seeing these metal sites in proteins and being able to use all kinds of energy and light to probe that and figure out more fundamental information about our world, that was what really compelled me. And then when I joined Ninian Blackburn's lab, I learned that you could basically use a very nice high-powered beam to get really cool information about proteins. So I'm going to center you a bit by saying a lot of this work is about copper proteins. And the interesting thing for us is that copper, at least evolutionarily, is relatively new to organisms. Obviously it's been around for millions of years in our systems, but unlike iron, which has been around since the beginning of life, copper and the mechanisms by which copper moves around in the cell is still not as well understood. And so one thing that's interesting is that copper, just by itself, a free atom of copper would never be found in a living cell. And metal ions can be very important micronutrients. We all need them, but in high amounts, they become toxic. And so you really have to make sure they don't go to destinations that they shouldn't. And so I'm just showing you, you don't need to focus in on anything really in this diagram, except to notice that there are lots of different ways that copper can be moved from one place to another in, for example, a bacterial cell, which I'm showing you here, which has a periplasm, sort of a protective outer layer. And then we have the cytosol here. And generally in bacteria, which I'm going to be focusing in on because it's a great model for wider, applications and other kinds of organisms, you can see that a lot of things are happening in this periplasmic space. And that's because bacteria really don't have a lot of protection for their DNA. And so you want to keep all of those metal ions that could be getting in the way or causing problems. You want to keep those out here. And so that's really where we start to see some really interesting strategies by the cell to keep copper going where it's supposed to be, and also in the host pathogen interface, which I'll talk about in a second. And so yeah, the question that drives me is like, what underlies this level of control in the chemical sense, in the thermodynamic sense? How are we going from one place to another? How does a metal ion move directionally from chaperone to target? And so we think about gradients or thermodynamics, or like, what is the driving force for something? And proteins are no different. Really, there's, we see what people tend to call big moosey creatures, these big crystal structures. I'm sure you've seen a few, even if you're not in biology. And then you might see a little metal ion in there. And really, that's the part for me that's the most interesting. And how is it going from one place to another, especially when There's another protein that looks basically the same in terms of the ligation sphere, and yet it will move from one place to another. And I like to think about it as this game of thermodynamic hot potato, where you just basically one protein wants it more than the other, it takes it away. The other has a slightly higher affinity, it takes it away. So normally that's kind of how this happens. And there is a scientific representation of hot potato. And this is a really beautiful graph that Lucia Bonci actually made, which is showing delta G. It's kind of, it's kind of one of those fancy three-dimensional graphs, but basically what you're seeing is up here, this is the copper-1 delivery pathway of sort of a general cell, okay? And if you want to pass copper from one place to another, say it comes into the cell and needs to go to a final destination, like that purple protein that I showed you at the beginning, copper A, how is it getting there? How is it installed? And we can see that you start with a relatively positive delta G with something like glutathione. And then it goes down basically to the next protein that would accept the copper, to the next protein that would, accept the copper, and so on and so forth, until you get to something called metallothionine, which is basically the rescue when you have too much metal in the cell and it's got to grab it up. So this is just showing a few different processes, like the metallation of COX-2 of the cytochrome C oxidase. There's a bunch of famous stuff here that probably might not be necessary to this audience, but these are actual processes that we have defined as getting metal from one place to the other in the cell. And so one way that humans have evolved to use metal ions is actually really interesting. And so don't focus in on the sort of busy thing over here for now. Look over here. And so say you eat a bad piece of lettuce. We hear about this. Don't eat that romaine lettuce. It's got a particularly toxic version of E. coli in it. And what happens is that E. coli gets into your stomach. It is able to fool your stomach and allow it to go through your epithelial cells and get into your bloodstream. And so this pathogen has the potential to make you very, very sick. And so our body will fight back by engulfing the pathogen in something called a macrophage. And then the macrophage will direct the pathogen to this killing chamber, literally called a phagolysosome. And so in there is where we can see this sort of war zone, okay? And so for different kinds of bad bacteria, here we see Streptococcus, Mycobacterium, Salmonella, and what I'm going to be focusing in on, which is E. coli. You can see that what is happening is all of a sudden when it gets in the macrophage, look up here, the macrophage begins to recruit a bunch of copper transporters to the area and begins to flood these bacteria with copper. And it's also been shown to happen with manganese and some other metal ions. And the thing is, at first what we thought is like you're flooding it with copper ions, maybe you get some free radical kind of thing going on, some Fenton chemistry. And that's true, that does have the potential to gum up the works. But really what happens in this phagolysosome is that copper, because of the Irving-Williams series, it's basically binding to any kind of metalloprotein that would normally bind, say, an iron or manganese or something else, and that gums up the works as well and kills the bacteria. And so what we're seeing here in this sort of war zone is the way that these bacteria fight back. And so I'm just going to point out for E. coli, that's what we're going to be focusing in on today, is the way that E. coli fights back against copper stress. And so there's a bunch of different, you've got like a sensor that's going to start, taking the DNA that codes for certain proteins, and then they start making the protein, and that gets installed and begins to fight back against this copper stress. And so I'm going to focus in on this big thing. That's the thing that you can see is different in this diagram, is this giant sort of system that is spanning the periplasm and pumping out copper. Okay, so that's really where we're kind of zeroing into, is this. And the idea of studying these systems is, I guess, long-term, if you could figure out how it works on the molecular level, on the atomic level, that you could perhaps find a way to shut a pump like that down without using antibiotics, right? And so that's not necessarily my interest. I'm much more looking at the structure and the fundamental chemistry, but people definitely could use this information in the future to deliver some kind of a deadly copper that could kill this pump. So I've put together a nice little cartoon of what this pump looks like based on the beautiful crystal structures that have been produced. And it's very affectionately called the periplasmic vacuum cleaner. So this is the CusC, or copper sensing system. And there are 4 components: CusC, CusF, CusB, and CusA, okay? So I'm going to interview our little casted characters here. So CusC is basically a big pore that just opens up into the extracellular space that allows the copper to flow out. So say we have copper stress, that's kind of what I'm showing with these little not to scale atoms of copper and silver, which I'll talk about. Then down here we have CusA, which is really the pump part. So during copper or silver stress, it's coming in, the cell can't, bacteria really can't defend against metal ions. It's an essential micronutrient. So it's just gonna let them in to the periplasmic space, but then that's a huge stress and it needs to be dealt with. And so this pump gets assembled. And so this down here, CusA in gold, is the actual pump. It basically translocates a copper ion for every proton. And so that just equalizes the charge and creates movement. And then over here in the middle, we have CusB in blue, which basically holds the two together. But as we'll talk about in a second, it actually has more of a role than that. And then over here we have Cuss F in green. I'll try to keep to the color coding for you, so just kind of keep track of that. But Cuss F is a metallochaperone that can carry copper or silver. And so when there is this stress, Cuss F is expressed in excess. So a lot of Cuss F comes out and it acts as a sponge to grab up these toxic metal ions as quickly as it can. And so what I want to point out is that the relationship between Cuss F, which is really the motile thing that can move around, and then CusB here, normally in these families of big pumps, something like this blue protein would not really be doing anything but kind of holding the pore and the pump together. But something that's very rare about this is that right here, this is an uncrystallized region. They weren't able to resolve because it moves too much. It's not just a fusion protein. There's a region right here that binds copper or silver, and it actually can undergo different conformational changes. And for a while, we really didn't know what its role was. And so that's what I'm going to talk about, is how we can figure out the mechanism of how this thing actually works by using XAS. And importantly, Cuss F and CusB were shown to interact. They could exchange metal with one another, but we really didn't know what each of their roles were. So at the point that I had come on to this project, there was a lot of argument about the role of that blue part. Really what people wanted to do was to fall into two camps. Okay, well, one group says, I think that when metal is in that periplasm and it needs to get out into that pore and basically impart resistance, that CusB is acting as an intermediary between the big business part of the pump and the chaperone, right? So that was the relay model, that silver or copper would go from Cuss F, the chaperone, to the blue protein CusB, and then CusB would pass that along to CusA, okay? So that would be the relay model of F, B to A. And then there was another camp that said, no, I don't think that's the case. That seems relatively unnecessary. We really need to get the copper or silver out of the cell as quickly as possible. Why would we want, sort of this intermediary? And so there was another camp that said that this blue protein CusB is a switch, that once it binds metal, that somehow activates CusA to accept metal, and then Cuss F, the chaperone, would quickly transport it over to CusA. So here we would be skipping B, but it needed to be metal loaded, and copper or silver would travel from F to A, okay? So there are three things that I really worked hard to figure out how to exploit to figure out this mechanism. And one thing I want to point out is that we are dealing with copper in the +1 oxidation state. So this is silent to most at-home or in-lab forms of spectroscopy. Copper too, you can get lots of beautiful UV vis, you can do EPR, but here we're talking about copper 1, which is the predominant oxidation state of copper in the cell. And we really didn't quite realize that for a long time. And there was probably a lot of mistakes made along the way as we had oxidized forms of our protein and we really didn't know. And so now that we are aware that we're dealing with copper 1, that adds an additional challenge to trying to track what's going on in these proteins. You can't just put it in a UV vis and kind of see the change. And so we had three main things: the chaperone Cuss F and the periplasmic adaptor protein CusB. they will transfer metal to each other, but only if one is empty, okay? So one of them has to be empty, and then the other one will come in and give the metal to them. So I knew I could exploit that for a reason, and we'll talk more about that. Second, the CusC system binds both silver 1 and copper 1. So one weird thing about this system is that the idea that it also is very specific to silver as well. And we really don't understand why, except to say often copper and silver co-mineralize in the environment. They may be present together. And as most of us know, hospitals, burn victims, we use silver 1 and we use copper 1 to actually fight bacteria. So it just seemed to be that that's the specific thing, but I can use that to my advantage as a second metal probe in my X-ray work. And then the other really cool thing, which is definitely all Ninian Blackburn's idea originally, is that we can use selenomethionine instead of regular methionine amino acid to sequentially label either one of these proteins, which gives us an additional EXAFS probe, okay? So these are the 3 things that we're gonna be showing in a minute. And this is the last slide before questions. So this is just explaining what selenomethionine labeling is. So this was actually first pioneered to overcome the phase problem in crystallography. And so structural biologists realized that if they could force the E. coli to take up selenium labeled methionine, which is an important metal ligating amino acid in many proteins, particularly for copper 1, that they would then have an additional way to do MAD and be able to solve that for phase. And so that was super useful for them, but not necessarily something that other people were using. And then Ninian, my PI, who was my PI, my former PhD advisor, he'll always be my PI, he realized that, most copper binding proteins have methionine ligands only in the active site of the protein and nowhere else, because they're actually very specific to binding and recognition. And so he said, let's incorporate selenomethionine, and it only will go into the active site of the protein. And so since you can see it's below sulfur in the periodic table, it actually just provides extra scattering, which is really nice. It provides a really nice probe. And we can switch edges and actually do the following. So right here is a lot of what you're going to see in the next few slides, where, for example, this is what it looks like at the selenium K edge when you have selenomethionine labeled Cuss F loaded with copper 1. And so you get this beautiful selenium carbon peak, which is a nice internal standard because selenium is seeing 2 carbons on either side of it. And so that's really a nice standard for having everything there. You've got your label, and then here you have that beautiful selenium copper interaction at about 2.4 depending on the protein. And so then you could incubate that with the acceptor protein CusB and see the selenium copper interaction decreasing in the Fourier transform. And so we actually can track transfer in this manner. And the beautiful thing as well is that you can then switch to the copper edge and see what the copper is seeing. And so we can see the ligation changes as we go. And so that's really the power of this technique. And as I'll show you in a minute for the question that we wanted to answer. So I had a blank slide. Yeah, here, we'll just go here for now, and I'll take any questions you might have.
That's fascinating. It's a great introduction. Thank you.
Oh, great.
One general question is, are there other cases in metalloprotein work where people have done this selenium substitution to be able to change the strength of backscattering from what would usually be sulfur? It's really wonderful. I mean, it's a great idea.
It really is. I mean, clearly the credit, goes to crystallographers at first for saying, hey, this is a way that we can have this additional scattering thing. But really it was Ninian who started using it for this particular purpose. And it was to actually probe the metallocenter of copper A, which is that first protein I showed you, which people have been working on since, I want to say, the 70s to try to figure out. I think Riti might remember from Ed Solomon's group some of their stuff that they were doing. And I think that Ninian, Solomon, and Fee worked together to figure this out, and it worked. I mean, it just kind of was this random thing. So we have noticed that people are tentatively starting to use it, kind of like they'll email us and be like, can we use that? And we're like, yeah, go for it. Of course, do it. And so people are now using it, I believe, in, God, like the active site of nitrogenase. They're doing some selenomethionine like insertion there to see what's going on with the various things. I know that we've done it for sure with some iron sulfur cluster stuff. But yeah, people are starting to use a little bit more, but they've always been cautious because it was sort of Ninian's thing. We're like, just go use it and see what you can do.
Okay, very good. There was a question, how easy is it to get the selenium in, and how much does it cost roughly?
It's actually not that bad. So the main thing, I gloss over the part of my job that is biological, which is raising these E. coli from cradle to grave, right? So there's a lot of cell culture. You got to get the DNA, you got to put it in. So what we end up doing, the selenomethionine is relatively expensive. You can get the racemic mixture. So all of nature is handed and chiral. So generally we'll only use one isomer of selenomethionine. And so you could buy the racemic mixture for cheaper, but it's only going to incorporate half of it. And so I've found that I buy the straight up, the D or the L, I can never remember, just whatever, dextrorotatory amino acid for selenomethionine. And basically what you do is you grow the E. coli normally for a day, get them nice and comfortable, and then you take them and you put them into minimal media, which is basically has no metals, has very, very little. The cells already have taken up most of what they need to survive for basal level. And then once they go into minimal media with regular methionine for a little while, just to get used to what that's like, then you put them into the media that is enriched with the selenomethionine. And it can be a little stinky, but it works really beautifully. You can mess it up for sure. So, but it's not that hard. It's not that bad. And if anyone ever wants a protocol, I'm happy to share it.
Just curious, are you going to talk about the fact that the copper bonding doesn't change the selenium to copper bond length?
Can you expand?
Well, can you go back to your Fourier transform?
Yeah, sure.
Right. So your first peak isn't moving when you
Right.
bring in the copper.
Right. So the first peak is you're talking about the carbon selenium.
Sorry, selenium
bond.
to carbon. I apologize. Yeah.
Okay. I was like, what?
My mistake. Sorry.
No, that's fine. For the most part, it won't change in the same system. So first of all, that carbon selenium bond won't really change much. And then if you have a couple of selenium labeled things, they might change a little bit. Yeah, it doesn't, the Fourier transform, the selenium carbon interaction will not change. And we find that to be actually a more valuable control to show that you have your label, and that when we did our normalization of the data, when we reduced the data, that we don't have some kind of funny side effect from what we did when we reduced the data. So you want to see it be pretty much the same. And, interestingly, when you combine the two proteins, you see that there is a little bit of attenuation there, but I think that's also just because you have more in the XAFS going on. So it's kind of a nominal thing, but I always really like to see it.
Okay. All right. Thank you. You should continue.
Okay. So this is sort of the experimental design. What I wanted to do with this process is that I wanted to rule out one of the models, right? I had said that there was this model that Cuss F would be a relay to CusB, or that CusB is a relay, that Cuss F, the chaperone, would move the copper over to CusB and then to CusA. So that would be the relay model. And I'm going to explain all of this, and I will take more questions if you have any right before we go to the next part so that you're really clear on it. So basically what I did is a series of experiments to rule these out. In the first, I loaded Cuss F. We're going to put them all three in the same test tube. We're going to mix them all together, basically activate the pump in vitro, hopefully. Okay, and so this obviously took a very long time to prepare everything. The silver is light sensitive. I had to work in the dark. There was a lot going on there. So first of all, like I said, we have the Cuss F that is loaded with copper 1. Great, that's going to be ready to go. So it is going to be the donor of the copper that we're going to track. CusB, I loaded with silver. That gives us an additional EXAFS probe. And importantly, I have the selenomethionine label on the CusB. Okay, and then we have Cuss F, which is a membrane protein, and it's a pain in the ass because it's a membrane protein, but we got it. And so here what we would see is if CusB was a relay, we would see a change in the copper-silver occupancy as it activated and the relay occurred of Cuss F moving copper to CusB, CusB moving silver to CusA. So we would expect to see something in the selenium EXAFS that would show a change from silver occupancy to maybe copper, or maybe just APO, maybe just empty. Something will change. And so that was what we did with that experiment. If it does not change, if we still see silver and selenium, then that means CusB is not acting as a relay or an intermediary. It perhaps instead is acting as a switch that once activated with metal is somehow activating this pump. And so that's what the second experiment is that I then put the selenium label on the Cuss F, okay, and loaded that with copper. Identical experiment, except that we have the selenium label on the on the Cuss F. Here we have the silver loaded CusB, but here what we would see is if this didn't change, so it's really dependent on this experiment. If this didn't change, then therefore we should see copper moving from Cuss F. That should become empty, and it should be moving over to CusA. And let me just mention there are a million controls that I've also done involving all of these, and I won't go through all of them. So if you have a question about did you try this, did you try that, please ask me because I probably did. So any questions on this particular setup before I move forward and show you some Fourier transforms? Okay, great. So we see what's going to happen here. All right, so the first thing was this selenomethionine labeled silver loaded CusB. And what I saw was the following. So what I was looking at was the selenium edge, although we could easily have gone to the copper edge or the silver edge, which I did. But essentially, here's what we see is that first what I wanted to do was incubate CusB alone with the pump. Okay, do we get transfer from just this guy to this one? No, we don't see really much change at all. It doesn't really do anything. It doesn't disappear. So CusB is not transferring metal to CusA by itself. That's important. And I know folks will have questions about the increased affinity of selenium for silver, but I will say that we have shown easily that there is transfer from this system. We can rip it out very easily. It's not just stuck or a dead end complex, okay? And so then when we mix the three together to test out this relay model, what we see is very, very little change in the CusB environment. So we see our selenium carbon, great. And then we see, it's a really interesting peak actually, the selenium silver peak. It almost looks like there's two things there, but in reality, it's just one. And I've always been curious about that. But basically, we see a little bit of a change in the shoulder that shows that some silver may have moved. And when we later look at the silver edge on the other, which I can tell you there was a tiny bit of APO selenomet CusB. So a little bit moved, but not very much. So there was a question, is this a dead end complex? Did we just make something that's not doing anything? Or is it perhaps acting as a switch instead of a relay? So we didn't see copper coming in. We didn't see anything changing with CusB. And so this brings us to the second trio, which is where we have the selenomethionine label on the chaperone, on the Cuss F. And then we have the copper loaded. And here we have CusB loaded with silver as well. And so you would imagine that if we have this switch model where CusB, when activated with silver or copper, it doesn't really matter, that then Cuss F is free to deliver its cargo to CusA. And that's exactly dramatically what we saw. So when we put these three together, so first of all, we incubated Cuss F with copper 1 alone with the pump and showed that nothing changed. So without CusB, without that intermediary acting as a switch of some kind, you don't get transfer from one to the other. CusB is actually really important somehow. And then when you incubate all three of them together, you immediately see, I mean, this is within the span of, the 5 minutes it takes me to load an XAS cuvette and freeze it, right? So this is happening pretty quickly. And immediately what you see, I actually can't really simulate any copper in. at all. And what you instead see is now that longer selenium silver coming in at the selenomethionine Cuss F silver. So what's happening is that Cuss F delivered all of its copper to Cuss A, and then some back transfer occurred from Cuss B to Cuss F. Because this was empty, now this guy was free to bring some over here. Now, we kept everything strictly stoichiometric, which is why we didn't see movement through the Cuss A pump or something like that. So we kept everything 1 to 1, and that is why we filled up Cuss A with copper, and then that left no choice but then this directionality with this communication between the two of them. We ended up seeing some silver go back in. Any questions on that for now? I'll have another break in a second, but yeah.
I think we're good for the moment. You should keep going.
Great, thank you. I just know for a lot of people, this is the area they're like, what? Okay. So then what's so cool is that we can switch edges, on all of these. So we did the selenium to be able to see what's going on. But when we saw that selenium copper interaction completely disappear in the previous slide in Cuss F, well, where did the copper go? And so what's so cool is that we can now go to the copper edge and see where it went. And so what we see here is an environment that I have worked with Cuss F and CusB and CusA, to the extreme. I know everything about what they look like in the EXAFS, I know what they do. And I saw a brand new site, something I had not really ever seen before. It's similar to what we saw when we just metallated this pump, but not quite the same, which I was really fascinated by. And so what we have is one copper oxygen nitrogen ligand. It wasn't multiple scattering. It's just some kind of a glutamate or oxygen or nitrogen ligand without any kind of a ring. And then we had 2 copper sulfur bonds that were consistent with methionine. And so Cuss F transferred its copper to Cuss A. And interestingly, this is the entry site of the Cuss A pump. There's a beautiful paper, and without the work of Sue et al., I mean, this just wouldn't have had we just wouldn't have had the support for this. But basically, this is the entry site, and you can see that at any given moment, there's generally 2 methionines involved, and then there is this glutamate, this oxygen ligand that's right here. And there was just a paper published this year in which they indicated that this was the preferred mode, was to have this 2 copper sulfur and 1 copper nitrogen oxygen. So we know that we're in Cuss A, and that we actually activated this pump in vitro, and that CusB is acting as a switch. And so, of course, you'd have to read the paper to learn more, but basically what we think is going on here at this point is that if the copper concentration is high, we're under copper stress, CusB will be quickly loaded with metal by Cuss F, okay, and that activates the pump, the switch, it does the thing. And then Cuss F is able to deliver to Cuss A quickly and freely and start pumping things out. Also, Cuss A may be able to passively just grab copper or silver and pump that out as well, but it has to be activated by this little region right here on Cuss B. And if the copper, you don't want to extrude all of your micronutrients. You want to keep some copper, you need it. And so that's where we think the communication between these 2 proteins is crucial, is that as you start to run out of metal, Cuss F will become empty and will start stealing back metal from Cuss B, thus shutting down the pump incrementally and basically in a more, a smooth flow, which is what nature is all about. And so that would deactivate Cuss B, and by proxy also deactivate Cuss A. This is just theoretical, but that's kind of what we were going for. And so what I'm next going to talk to you about but I'll take questions first, is the rapid freeze quench XAS that we did to try to figure out more of this relationship between Cuss F and Cuss B. Why are they so paired with each other? What are they communicating? And how can we investigate a copper-1 system in real time between 2 proteins? And that's what I'll talk about next.
Great. The experiment design is wonderful. The ability to select which of the proteins you're actually interrogating for the presence of copper versus silver. That's just great fun. You showed the actually we have a question. Yang Ha, would you like to ask your question?
Yeah, sure. So I assume a lot of ligations of such ketone ions are mainly dominated by the steric effect. But in copper 1, I know when it binds with sulfur, it can have some maybe 5% back bonding. And I wonder if you have any experimental or computational data show the difference of the back bonding in copper 1 and silver 1. And do you think that caused the intrinsic difference between the binding behavior of these two?
That is always the question. I love that question. So for sure, silver and selenium. So first of all, the edge data is a little bit crappy with this, and I'm not much of a Zanes person. So actually, if anyone ever wants to take a look at the deep Zanes of this, I would love to know more. We generally will pay attention to the copper one, just sort of at the edge, because it'll tell us a lot about coordination as well as the extent of that kind of back bonding, but it's not as informational as probably going further into the Zanes, which is more used for copper 2 in our field. But anyway, we definitely see back bonding into the copper. We did some XES on this, actually. There is a paper that we have. And in fact, I think might have been some of the first people to do that with Demosthenes and, I can't remember his name. Riti, what is his name?
Thomas,
but then there was a person before who went back to work in China, I think. Su Chen. Yeah, I just wanted to make sure he got proper credit. But anyway, we took a bunch of proteins with selenium label as well as sulfur, and we actually went through and looked at these interactions. And I am not an XES girl. I was more the biologist in the room for that one, I gotta tell you. But the paper itself is very robust, and I point you toward it. Maybe I can put it in the chat in a minute, and that should answer your question.
Just a follow-up question. What about zinc too? Have you run any tests on that?
Zinc will not bind to this protein.
Okay.
But you're absolutely on the right track to think that would be a good thing. It's just actually remarkable how specific this thing is, how evolved it must be to really only bind copper and silver.
Cool, thanks.
Okay. You should continue, please.
Okay, how are we on time?
We're great. We're doing just fine.
Great. So this is kind of the part I like to call a secret handshake, okay? And it's really the part where the plot sort of thickens and it becomes a little bit more challenging in a different way. I would say the last bit was very, very challenging because I just had to make sure everything was perfect. I mean, how do you make sure that you have fully loaded protein? How do you make sure that you have perfect stoichiometry? How do you make sure that the labeling, all of that working it was truly a labor of love. And we did it in duplicate. We did not just do it once. And so that was really, really important to us, and tons of controls. So then we start to wonder, though, what about this relationship between these two? Why is this so important? And as we started to explore this, we, ran into this problem that Cuss F and CusB have literally nearly identical copper 1 and silver 1 binding affinities. And so, yes, I said that if one of them is empty, the full one will transfer its metal over to exactly half, and then they'll stop. If both are full, they don't scramble. We call it scrambling. They don't really exchange metal or anything like that. And if you look at the active site ligands, so what we have here for Cuss F, it's a pretty small protein. It has 2 methionines and a histidine, and it has a tryptophan that I'll talk about in a minute, okay? It's the only tryptophan in the protein. Over here, CusB, this is the N-terminal little fragment that we're working with here, which we've done enough controls to know that that's enough to do what we want to do. And it has 3 methionines. But when you rip the copper out of these, they have the same KD. They have the same cave dissociation. And so there's really no thermodynamic gradient, which is usually what defines everything. We think about it in terms of, we have met-met-his and met-met-met, maybe having 3 methionines is going to draw it over more. But in fact, all of our experiments show that that is not the driving force for this, that they actually have no difference in their desire for copper, which is against nature, you know, at least on its face, because usually, like I showed you, remember I showed you that delta G diagram that showed one protein has a more positive delta G than the next one, which is more negative, more negative, more negative. These have nearly identical delta Gs, basically, of binding copper. So how do we do this? How do we understand the nature of transfer? Kinetics is the story. We need the kinetics. And how do we do that? If we have copper 1, silent to most fast forms of spectroscopy like stop flow or something like that, they have a very shallow thermodynamic gradient, similar binding sites, and are pretty much impervious to a lot of more straightforward methods. And so again, just reminding you that we're going to be using the selenomethionine labeling here, but in a slightly different way. So I started to work with, in my final years of graduate school, I started to try to come up with a method for reliable, rapid freeze-quench XAS. And so how this works is really just, it's like working on a car, right? But you don't really know what the car is going to look like when you're done. You're creating a method that is unique to whatever the project is that you're working on. So what we have here is Stewart, which is my rapid freeze quench in my own lab. So when I left graduate school, I hadn't quite figured it out. And so when I got my own lab at Reed, I got some funding from the Murdoch Trust, which is up, it's for, and I also want to point out in an undergraduate, primarily undergraduate institution. So this is where I brought in a lot of undergraduates to enjoy the beam. But basically I got a nice little grant from them to work on this arm of the project. So Ninian and I are collaborators, but I ended up sort of taking this on as my own pet. So what you can see is there are 2 syringes. We have reagent A, which could be Cuss F loaded with copper. And then we have reagent B, which maybe would be labeled CusB, empty, ready to go. And then you put them in rapidly with a servo, you push it really fast, and you have nearly perfect mixing, if you understand what that kind of means. And then it goes through an aging loop. So that's where we get our time resolution from about 13 milliseconds to infinity, okay? And so then what we do is we spray that mixture, you see my student made it orange, of Cuss F and CusB in real time, whatever they're doing at 13 milliseconds, all of a sudden it's quenched by liquid ethane, which I produce by basically putting a tube of ethane gas into a little finger in some liquid nitrogen. And so I make that, and what's nice about liquid ethane is that it sublimes out of the sample. So you have this snow that's frozen, but unlike isopentane, which is kind of oily and will dilute your sample by 50%, this just goes away if you put it in a -80 for a minute, for half an hour, and then I can stuff it into our XAS cuvette. And at first we were worried about not being completely homogeneous. That's what you always remember, right? We got to grind up our samples. We got to make sure it's completely homogeneous. But because we're doing this fluorescence, and because of the way that we designed the cells, we actually really didn't see any internal absorption. It just really was, we got really lucky. I mean, it took some time to make sure it was all working well, but it really has worked well. And so what we end up getting is kinetic and structural data, okay? So this is what you're looking at, and I'll point out some issues with it in a minute, but this has been done, I did different temperatures, I did different stoichiometries, I repeated these experiments many times. And so what we're seeing here is not just a one-off, let's see this kinetic, thing in XAFS. It's just, reproducibility, going to the beam. I don't live at the beam, so, I've got to go so this took quite a while to see what we're seeing. But this is what we ended up with and ended up being able to replicate over and over, which is to make it move a little quicker, we have a 2-to-1 ratio of selenomethionine labeled Cuss F, and then copper 1 loaded CusB. And so what we're looking at here, instead of the decrease in the selenium copper peak height, we're looking at the increase because it's going from a non-selenium labeled environment to a selenium labeled environment. And you can do it either way, and we did do it both ways. And so what you can see is that, yes, we look at the Fourier transform amplitude just at first and say, okay, what is increasing or decreasing? And immediately what you can see is we did not get what's most important here, which is the earliest time points. But over and over, we got the same profile of this very fast, immediate transfer of metal, and then sort of this slow transfer after that, okay? And we saw this over and over. And so we were able to get a couple of kinetic parameters here. But what was more interesting to us, yes, we got it. That was really fun and you know, challenging and interesting. But right around here is where you're going to have these two proteins sort of locked together. They're in complex at this point. They haven't gotten rid. They're still hanging out. And so what we were able to do is then take each of these, you know, these plots, these EXAFS that resulted from each of these. I'm just showing Fourier transform here. And we can actually go into and take the copper edge, for example, and the selenium edge and be able to see what ligands are coming from where. The secret handshake, okay? Because this is what we are beginning to understand about these low thermodynamic gradient proteins, is that the two proteins will come together and they will begin to sample ligands until the perfect little setup somehow, they reach some little thermodynamic minimum and it takes it away. And so basically, depending on which protein had the label, we could actually predict, okay, where are the selenium ligands coming from, okay? And that told us kind of what was responsible. And so depending on where the ligand was, or depending on where the label was, we either saw one methionine coming from, sorry, I'm trying to see around my video here. We had two populations. One population, when they locked together, had two ligands coming from CusB and one ligand coming from Cuss F. And then the other one was a second population where we had a met and a histidine coming from Cuss F and then a methionine from CusB. It's not too important, but just understand that we can actually see kind of that there are these two populations that would happen. And the fact that the label did affect that was actually useful to us. We did see a difference in how quickly selenium was copper was transferred, but that was helpful to us in this case. And so we had Kenny Mers, who does some really cool in silico work. Independently, he had published some papers on what he thought was happening just purely in silico. And then we showed this data and we talked about this data. He said, that's so funny. I have two predicted populations as well. One where the CusB is dominating, basically has more ligands involved, invested, and then a minor predicted population where you have one met from CusB and then the rest is coming from Cuss F. And so we didn't know that at all. He was like, this just kept popping up. So it's super freaking cool that that agreed with our data. like we just never get that to happen. So great. We've got this, we've got all that information about the nature of that connection. But the problem is, I don't care how good you are, you can't go to a publish, with your manuscript and be like, this is our kinetic curve. It's not. It's really showing transfer in real time, but this is not a kinetic curve. Like, what did we even have to do to fit that? We did like some weird biphasic thing. So this was great to know, but we just needed to get something quicker than this. It's just really fast. So we need to use a different vantage point. And I'm going to go away from XAS for just a brief moment to show you how we did this, because I think it's really cool. But it complements the data. So any questions on that for now? We're almost through, so if you want to wait, we can.
I think we'll hold for questions at the end.
Cool. So this is where we can use tryptophan. So I'm sure many of you know, but just so people do know, is that you can actually look at the intrinsic fluorescence of a active site adjacent tryptophan. You can see solvent effects. And in a lot of cases, I don't know why, and nobody's really talked much about this, but the more I've been talking about this work I'm doing, the more people have popped up and said, hey, my crystal structure for my metalloprotein shows a, aromatic residue right next to the active site too. Maybe I can use this. And so there's something about these aromatic residues in active sites of metalloproteins. It's doing something, but it's super handy to us. It's a beautiful spectroscopic handle that is not a tag. It's not something being added on or changed in some way. It's inherent to the protein and it has a really high fluorescence yield. So Cuss F, what we realized we could do is that Cuss F has that little tryptophan, and it's even been called a cap, although it hadn't been proven at the time or really shown. But it does seem like it's doing something. And so we found a paper where someone had titrated in copper 1 to Cuss F, and you're looking at the fluorescence here. So you can excite at 295, and then you look at the emission at 350, which is quite large. And basically what we showed is that we could titrate Cuss F with copper 1 and get a response that was very, striking and a good yield. Like we could get a good signal on this, and it was 1-to-1 with copper. Great. So this tryptophan is reporting on what's going on with metal binding in this protein. We have another tag. Interestingly, because I didn't know much about fluorescence at the time, I took it out past where we were interested, but before the first harmonic, and I ended up finding this little cation pi peak here, which is super interesting for other reasons. But that's a cation pi interaction between the copper 1 and the tryptophan. And so we can use either one of these peaks to track copper at home, in a lab, not having to go to the beamline with copper 1. So it's a nice complementary thing, but I will never stop wanting to go to the beamline. So we can use this to show protein transfer or metal transfer in proteins. So this is showing CusF empty with that nice big signal of tryptophan. And then when you incubate it with copper 1, that disappears. And then if you add CusB, we can do the experiment where we show these two proteins and you get the same thing. So we can sufficiently track metal binding using just tryptophan fluorescence. And good old Kenny Mers to the rescue showed us what he thought might be happening with that tryptophan cap when CusB and Cuss F are together. So what I'm about to show you is the real-time stopped flow that I did, and I'll show you the setup in a second. But really what you'll be watching is the quenching of that fluorescence as the cap is taking on copper 1. So we're going to look at things from Cuss F's point of view, the chaperone, as it takes on metal from CusB, that quenching event, okay? So this actually took me, again, a lot of years to figure out. You can do fluorescent spectroscopy in a stopped flow, which is a lot like the rapid freeze quench I showed you. 2 syringes, they mix rapidly, but instead of going into snow and into a little vat, it goes into a cuvette. And then whatever you want, you can shine whatever light you want on it and get whatever light you want out. So you can do I have a PDA, a photodiode array I can use for UV, but instead what I had to use at first was this sort of dinky photomultiplier tube, and you could only get so much, and you had to use filters and all this stuff, which is great, but I talked to the company, ended up getting an LED source that will excite just at 295, giving us an external way with tons of photons, and then that goes into the stop flow cell, and then I rerouted everything so that instead of doing a bunch of filters and sort of windowing in what I wanted to see, I had the monochromator do that for me instead. And so I selected one wavelength, which is that 350 peak, because it's so bright, and then that could go to the fluorescence PMT and give us counts. And so I just kind of moved things around. The caveat is you only get one wavelength, but the signal is super clean, and I don't need to use a bunch of filters. And so again, what I'm showing here is basically this cap. That's what you're going to see in the next image, is how this is happening. So we're not seeing in the same way we saw the XAS or the Fourier transform. That's physical metal. We can see the metal doing what it's doing. This is indirect. So I just want to make that clear. And after a lot of playing around, making sure that fluorescence can be junk. And especially when you're mixing at really high pressure, that quenches quite a bit. And so you could be looking at just complete crap. But after a long time, we figured it out and we caught that fast phase, which is super freaking fast, 165. Talking about a physical metal moving from one place to another, this is within diffusion. I mean, it's really fast in our neck of the woods. And then there's also this slow slope, which matches up with the slow sort of increase that we saw in the RFQ. So we were able to supplement what we had at the beamline. Everything that the in silico studies said was happening is happening. And then so what we see is that we have this initial rapid complex formation. And then after that, we have a slow release of copper 1, during which the tryptophan is allowed to fully go. So basically this is happening very quickly, and then this will happen over quite some time. So there's something funny happening. And then this is also just talking about how we can do and look at the kinetics for the selenomethionine labeled as well. And that helps us understand more about what we do in the RFQ, answering the question about back bonding and things like that. How much is the selenomethionine label changing things? Actually not that much, but it does change it. And so, yeah, I think that's all I wanted to talk about. I'm happy to answer any questions, and I just want to thank everybody on this screen, and money and all of that good stuff. And thanks for having me.
Excellent. Thank you. That's a great story. I really like that. We've got questions starting to come in, but I'll start with one. You had much earlier, you had the copper XAF showing that the second neighbor selenium had a double peak. Am I remembering that correctly?
Let's see. You tell me when.
Oh, it's back quite a bit when you showed the copper XAFs, and you mentioned the
This
shape.
guy?
No, I thought it was a copper XAF, so that
Oh,
showed
sorry.
that looked like there were two distances to the selenium naively.
Okay. I'm sorry, I got my big screen here. I'm coming. I know where you're going. Okay. Let's see. So over here? Over here? Over here? Sorry. I don't know where it is.
That's okay. In any event, it looked like you had two distances to the selenium, and I was wondering if that was indeed the case, or if we needed to think a different way about the copper to selenium.
Yeah. God, I wish I knew which slide. Just a minute.
That one. Oh. That's it. Sorry, it was selenium to
silver. Yeah.
I apologize.
That's great, because that's what I was thinking to myself, and I was
sorry.
No, of course.
I knew I wanted to ask about it, but then I didn't write down my notes about what the elements were, so I was guessing. Bad on me.
And I'd be interested to know what someone like Riti would have to say about this, because doing silver was not trivial, just because it's pretty high energy. So we were up at, I want to say, 23 or something like that keV, and so
this is the selenium you're showing us now. Did you also do the silver?
Right, I did do the silver, but what
okay.
I'm trying to say is that we hadn't really seen much silver Fourier transforms or XAFs. Even selenium silver, all sort of bets were off in terms of what we, you know how it is. You look at an XAFs and you go, oh yeah, that's got, this kind of pattern, and I know we're going to see this kind of like, we really didn't know what to expect. And so this was one of the weird things about selenium silver, is exactly what you said, this sort of double peak. And one thing that was interesting is when it was 1-to-1, we would see the two sort of peaks here. And maybe that's because it's in equivalent methionines, right? Maybe there's just a slight difference in that envelope of different states. The Debye-Waller itself was pretty large, and so I think that does sort of jive with that. And then what we would see, though, interestingly, is as I didn't do it perfectly every time, right? Sometimes I'd have kind of when I was doing the controls of silver-loaded CusB, sometimes the stoichiometry wasn't perfect, and maybe it would be 0.5-to-1. And what I would notice was this peak would completely disappear over the course of losing silver. And then we would just be left with this here. But when I simulated it, it all said it was the same distance. So, it's one of those funny things that sometimes does happen. And I think it has to do with, like I said, maybe these aren't quite, there's a couple different states, something like that. But we wouldn't be able to tease that apart with the homogeneity of the system.